Hopefully you didn't miss me because we were away. Thank you very much. And let's see. Did you receive? The reason I'm asking is we had a really poor Wi-Fi. So did you get the recording? I went out of sequence for a reason because when I do proteins and nucleotides, which I'm doing today, and perhaps some carryover into Wednesday, I'd rather be demonstrating it to you rather than blabbing about it. So I did a little introduction to cells and some of the roles and things like that, just to try to be where you are. And I'm sure you covered some of it. You probably did cell stuff in lab, right? Yeah. No, yes. Mission. Yeah, I'm not. You're not in tears over lab this morning for whatever reason. I'll have to talk to Dr. Robinson about it. See if we can work on that. But we digress. Moving on. So you're the one. The other groups, for whatever reason, I got through this, and it's so hard to try to get at least I can recall it briefly. This is something that is more relevant. Steroids are more relevant to you when you start to get into the endocrine system and really into the physiology aspects of it that are there. But they're extraordinarily vitally important. I mean, you saw a little bit if you got a chance to look at the cell. Cholesterol is embedded in every single membrane. Okay, our membranes, cholesterol is a stabilizer. And so it's an integral part, and there's a fairly substantial percentage of the membrane's cholesterol to start with. That's the molecule of cholesterol. Its other uses are to create what we call steroidal, and that's where it gets its name, the sterol. A sterol has a similar nucleus to that. This is basically the sterol nucleus here, hence the term steroid. Okay, and so those four interlocking carbon rings are the critical piece to recall. Cholesterol, all eukaryotic cells like ours, all have sterols. Okay, every animal cell has cholesterol in it. Okay, so if you take something else that's in that family, for instance, fungi, mushrooms, okay, the mold, they have sterols also. It's a different sterol. Okay, plants have sterols, but it's a different steroid. And you'll see down the road, if many of you are going on to micro, why that particular sterol in fungi is a target we use to address fungal infections, both orally, intravenously, topically, etc. Just to give you some idea. But really what we do, other than the stability part, we use this because fats, again, because they're not bonded together, we actually have to use a fat to separate it. Okay, and that's when you think of what a soap is, because we have dirt and grime on our hands, it's mostly fatty in orientation or in science. And so what a soap is something that is water soluble, so you can wash it away, and fat soluble, so it can bond to that, so you can wash it away. So effectively, there's a lot of uses, and when digestion, we have something called bile salts. And bile salts are something we recycle the cholesterol, a great deal of it, and we'll get to this, and you get to that in digestion. So that's sort of a little introduction, but this piece, we lop off a lot of these side chains and we make important stuff like estrogen, progesterone, testosterone, a variety of aldosterone. They all have that like last own sterone to show you what they're like. And I mean, if you know somebody who had a shot of cortisone, an older relative, because their knee was, grandma's knee was acting up, the doctor gave her a shot of cortisone, same deal. That's made in a different part of something called the adrenal cortex in the adrenal. Very, very important, bigger player down the road. So cholesterol gets a bad rap. Most of the bad cholesterol that we get is excessive amounts that we derive from sugar, and it's not so much that the cholesterol is wrong with it. When we talk about bad cholesterol, it's really fats and fat-like substances that we have an overabundance of, and the way they're packaged. Is such a way that we have they create mischief. It's very, very nasty mischief in the body. And then lastly, this is an eicosanoid. Eicosanoid is another name you deal with in AP2 when we talk about the immune system. The key word is here, prostaglandins. Okay, they got that name not because the prostate's only in men. It's not like women have them. Also because they were first identified in the prostate. From the prostate gland. but they're a pivotal piece in inflammation. And indeed, we activate them in such a way when we have inflammation, when you sprain an ankle, fell and cut your knee, whatever, or anything like that, get a splinter, anything that would happen to you, minor or otherwise, you have a virus or something like that, or a cold, all of those will generate inflammation because tissue is damaged. Chemical signals go out, and what happens, those chemical signals activate prostaglandins. And it's a fancy name we call pro-inflammatories, what everybody was talking about during COVID. You know, it generated a lot of these in overabundance, the initial presentation of COVID-19, not so now, comparatively speaking. Nevertheless, I don't know, I blocked my prostaglandins this morning. Every morning I take a couple of Aleve. Okay. Maybe you take ibuprofen. Maybe you were in a game or in a match or something like that over the weekend. There's nothing, you have, for whatever reason you might be taking it. Or maybe just take an Excedrin or an aspirin or something like that. Tylenol doesn't qualify. They are called anti-inflammatory drugs because they block the actions of prostaglandins. Okay? So they have a fancy name, non-steroidal anti-inflammatory drugs, or NSAIDs. And then when we previously looked at cholesterol, those are cholesterol-based or steroid-based anti-inflammatory drugs. They do the same thing. Neither one is, there's nothing perfect about anything like that. Tylenol is a little bit different. It reduces fever and it reduces pain. It has its own subset of problems. It's highly liver toxic in excess, for that matter. So many of you, as you go on to take, and many of you will be going on to take pharmacology courses, that's where all of this comes from. So effectively what they do, and maybe you know somebody had heart surgery. And what do they take for that? A baby aspirin a day, because the aspirin blocks enough prostaglandins so we don't get irregular blood clotting. Voila, that's everything. So the last little nugget for this, just for fun, didn't I tell you arachidonic acid? That's what we call an essential fatty acid. You have to have those. It's in peanut butter. Let's hear it. There are certain things that are important. I always knew peanut butter had value. Voila. On with the show. This is today's show. It's called Proteins. And if we get to it, and I hope we will, we'll get into DNA and RNA. So the show is about proteins. I say it all the time. Genes tell you what proteins to make, and you are what your proteins make. That's how we differentiate. There's a very limited amount of difference. About 97 to 98% of our DNA is the same. A lot of it's DNA that we call regulatory. Years ago, they used to call it junk DNA, which is the stupidest thing I've ever heard. Okay, but effectively, it plays a variety of roles, but really, there's a very limited variance between anybody in this room, okay, with regard to our genetic material. They are the most varied functions. They do structural ones. You learned about them in connective tissues, collagen, elastin, reticulin, and others. Okay. Every reaction, even this one we're looking at, requires enzymes. Muscles are nothing more than masses of proteins. They're masses of proteins that are accumulated. They're just packed, little cell sections that are packed and bounded in rods, effectively rods, and modified rods of protein. They're just packed super tight. A whole bunch of mitochondria for energy, and a whole bunch of other sources for energy, and oxygen carrying capability with other materials that you would learn about when we look at muscles. So they're all a little bit different. Carbon, hydrogen, oxygen, nitrogen, yes, sometimes phosphorus. Sulfur is a big player when you get to structure. That's why they put the S here. So you can think sulfur, think structure. The structural proteins, which are the ones, and basically structural proteins are pretty unique because they don't dissolve. They're insoluble. That's where they get their strength from. Nearly all of them. Not all, but nearly all. And they're held together by something called peptide bonds, which we'll look at in a second. That's on that side. And 4 structural levels. Voila. So we will look at these structural proteins. You're going to see, and I'm going to demonstrate it for you, not well. Sure. I go to a very, very low expense. I'm using paper towels. I'm going to get reimbursed for it. Therapy. Collagen, what's that? Braiding. Biologically, when we want something that's insoluble and very strong, elastin and reticulocytes. Enzymes, it's about shape. All of these. Because enzymes, again, they're not drawing at the scale. Bring molecules close to each other and accelerate the likelihood that they're going to bond and do something, either come together or come apart. Four different what we call polypeptide chains. In the middle of each of these chains is an iron compound bonded to organic material called heme. These lighter color are called globin. The heme is where the oxygen is. Just gives you some ideas what they're able to do. They're remarkable in that regard, I think. This is when you get to muscle, oh, you'll be sick and tired of this. This is called a contractile. Okay. Protein, it changes shape. So when you have something that changes shape, okay, you can create movement effectively by changing shape, attaching itself to this kind of pulls this upper leading glue called actin. Here's the one called myosin. It kind of pulls. Both of the actins are attached to the side boundaries of the subunit of the cell you're going to hear about called the sarcomere. And effectively, it pulls the walls closer together of that unit. In doing so, when you add it up, it creates movement. Muscles don't bend, they don't contract like you're folding. Okay? It has to do with their shape and how they contract. Some bulge, I would do that, but I can't. You would help me. As a human. I know it's early. I could do this, but I can't. Body by whole. Communication, transmitting signals. Best example in the world, insulin. Insulin basically attaches to the outside of the cell. It causes that protein to change shape and it generates a chain reaction on the inside of the cell. It isn't all about just open the door and glucose, like a lot of people think. No, insulin has a myriad of activities. Insulin causes cell growth. Insulin leads to multiple cells. And it's interesting, it uses sugar as its energy source. Down the road, when you hear about human growth hormone, it works very similarly, but it uses fat as an energy source. And indeed, the two of them have like a very interesting biological tug of war. When we're growing, we utilize fat. That's why it's the best of the performance-enhancing drugs. In a sense, create more muscle mass and reproduce cells, use fat in the process. Insulin uses sugar as energy. So we're more designed to do it a whole other way. Here's the defensive capability. There's a virus, the naked virus, it's called that. We learned down the road that COVID was a good example. It's probable it's called an virus. These are structures. So the antibodies basically lock on for various areas, immobilizing it and creating an identity that your white blood cells can come along and gobble it up. Fascinating. Okay, so the proteins are made from 20 different types of amino acids. If you were in that class over there, the ones meeting was like same voices, okay, you would have to know all 20 of those amino acids. You don't have to know any. How do they vary? Okay, we join them and you're going to see a very, very strong covalent bond. Very, very strong, usually strong covalent bond called a peptide bond. Here's what they look like. Okay, they can act as either an acid or a base, and they have 20 different R groups. So this is it. It's got a central carbon, has a hydrogen off of one end. It doesn't matter. Remember, these are three-dimensional. It has an acid on one side, one of those carboxylic groups. We just say acid for the time being. And here's a new player with the nitrogen. It's called an amine group. When you ingest a lot of proteins, you end up releasing a lot of these amine groups. They create ammonia, which is a very nasty toxin. One of the problems with people who overindulge with proteins, okay, or drink a lot of protein supplements, they've got to be very careful to hydrate to get rid of that extra stuff. And if their kidneys aren't working properly, that's a big problem. This side is basic or alkaline. This side is acidic. So you have effectively a neutral molecule that has an acid on one side and a base on the other side. Very unique. They're my horrible handwriting. So the big difference is the R group. And the R group could be as simple as a hydrogen, or as complicated as some massive amount of chemicals. Really, but effectively, if you want to identify them, you have ones that are either, the R groups are either acidic, basic, positively charged, negatively charged, water soluble. Simplistically. Indeed, all those first words, it's got an acid or a base pointing out, or something that makes it positive or negative sticking out, it's going to be water soluble. Anything else will be fat soluble. And that comes down and we start looking at the properties. So that's the starting off point. So 20 types of amino acids joined by a covalent bond called a peptide bond, act as either an acid or a base, either side, and 20 R groups. Voila. My bad artwork is over there. I drew it. See it. Once again, you split off water. So what happens is we take effectively this hydrogen and hydroxide that are close together, and we form a new bond. Take that away, split off the water, plus we go over here. We create a bond, not on the scale, as I do, between the carbon and the nitrogen with a C double bond O and H. Back. Interesting, and if you wanted to break it apart, again, you need enzymes to put it together. You need peptidases, different kinds of peptidases you'll learn about in digestion. And basically what they do, if you have the enzymes, split off the water, we'll make the peptide bond. We have the right enzymes, break it apart with water. Our digestive tract absorbs amino acids. This is called, we have two of them together, it's called the dipeptide. See, to me, I would think two peptide bonds, but no, I didn't write this. I've asked them, they won't change it. People in chemistry will not change it for me, the biochemist. Dipeptide means two amino acids, one peptide bond. Tripeptide means three amino acids, two peptides. That's why I don't want to ask you, because you'll start throwing you, which I understand. So I was throwing things at the set yesterday. I have one of those foam bricks. My children don't want to be around me. Thankfully, I was in the car for a lot of it. Okay. And here it is. 4 levels of structures. Everything has to do with what's called the primary or linear sequence. You have primary structure, secondary structure, tertiary structure, and quaternary structure. 4 of them. That's right. Count them, 4 of them. What is the primary structure? It is effectively a batting order. And just for fun, typical batting order. You don't have to name them or anything. They could be all the same. They could be all different. You can have 2 the same. That's why out of those 20, there's an infinite number that you can have because there's no limitations. 1, 2, 3, 4, 5, 6, 7, 8, 9. Make it easy because I'm running out of room. Okay, so that is called it's a linear sequence, and you'll see it linearly in a second. That's it, the primary structure. Again, because the carbons don't line up, even though there's angles that are there, effectively you have a number of these peptide bonds. You can see the carbon and the nitrogen, the carbon and the nitrogen. This is a sequence. And so effectively, once you get to like 4 or more, we call it a polypeptide. What's the difference between a protein and a polypeptide? Not importance. Oh, proteins are more important. No. Length. Proteins have 100 or more, okay, 100 or more amino acids. That's all. It's not something magical at all. It has nothing to do with their importance. It had nothing to do with where they're found. It's just a matter of size. It's like you have a big polypeptide change and a short. They just gave the name proteins if you have 100 or more amino acids. Okay. So that's the way it looks like. And we'll get to what happens afterwards in a second. The secondary, how they interact with each other. It's all about hydrogen bonds. Okay. Depending on those R groups, there's always going to be hydrogen bonds. If you go and look at this, you're going to always have an abundance of NHs here, and depending on that R group, okay, NH is here, and so you know you're going to have hydrogen bonds, and whatever that R group is, it's going to most likely have a lot of hydrogen bonds. And depending on the sequence of those R groups and all those other properties that are there, it's going to lead to something that looks like this. It's going to form one of two structures. It's going to form something that's called a pleated sheet. This is really important for you folks who are just studying about anatomy. I've been studying anatomy for more than 50 years. Okay, what we do in the body, and you probably heard me say it before, when we want to create space, because we have limited capacity in this body, we fold, we pleat, we fan fold. These are called pleated sheets. Accordion pleats, whatever works for you, one. Except they're all attached. We do a lot of that. The other thing that we do with these is we create what are called alpha helices. These are beta pleated sheets. You can see the script of beta. Okay, and the others are helix. Alpha is a right-handed helix. I don't worry about the handedness, but it's just a twist. We do a lot of that. When we get to DNA and RNA, they twist. It's a double alpha helix. So you have a combination of pleated sheets and alpha twist. And just as easier for me to do the twist, so I'll do it this way. You have an extra one. For fun, I'll make it into a pleated sheet. Again, I go to literally no expense. Voila. And in some way, shape, or form, they come together. Okay? What you can see here, this is called the tertiary structure. So, the secondary structure, and I'll go back. See those, wherever you see those little dots, structurally, hydrogen bonds. The beauty of hydrogen bonds is they're easy to attach and they're easy to come apart. Remember, it's a weak attraction. So you can see them all through here. Here, you have those ribbons that are attached by hydrogen bonds. Here, you can see that the hydrogen bonds holding, and it's all about the primary structure. Tertiary structure. So what they do is they're saying here on ribbons, which are the pleated sheets, and on the spirals, they formed a compact globular molecule and held together by intramolecular bonds. That means secondary structure is all about hydrogen bonds. Tertiary is this. Acids and bases seek each other out to neutralize. Positives and negatives attract. Water-soluble things go with water-soluble things. Fat-soluble things go with fat-soluble. We get two membranes. The reason we have what you saw before. Nice illustration, might as well use it here. How does that stick in the membrane? Because this portion is fat soluble and it's water soluble on the outside and water soluble on the inside. So something like insulin's outside. Too big to get giant protein. Effectively, it's like someone knocking on the door. You're going to do something. You're going to come and answer the door. You got to throw something off. You're half undressed. Who knows? Whatever it is. And it causes a change on the interior. That's what happens. The change of the interior. Remember, the exterior of the cell is water soluble, bless you, and the interior of the cell is water soluble. So how does a giant protein, it's not going to scale, the protein is giant size, how does it get in? It causes an alteration, like I said, a chain reaction. So one of the things that insulin does with one of these proteins, and I'll sort of simplify it, by taking one of these, basically the door is closed, but when insulin attaches, it opens the door remotely, so to speak, and boom, creates an opening that glucose will get in. When the insulin's not there, it closes up. So down the road, you're going to see it's something that facilitates or enables a process called diffusion. So we call it facilitated diffusion. It's this when we start talking about movement of membranes. That's what I'm trying to put the package together for you. It's hard now because you're not seeing it in its full throttle, but hopefully you will. So that's just the starting point with this. So basically, you get to this combination of pleated sheets and alpha helixes come together, and the tertiary is how they interact based on their R groups. And then frequently, not always, but frequently we end up with a quaternary structure. And just like hemoglobin, which is the classic example, okay, polypeptide chains, this one has to do with how transthyretin, functional proteins, it's got two polypeptide chains, you have the number of amino acids, and it assists with carrying thyroid hormone. Come to learn that thyroid hormone is fat soluble. And you look at this when you do endocrine, and it has to have a carrier molecule. And proteins, because a lot of them are water soluble, and they have areas that are fat soluble, will carry fat soluble things around. Fats move around in your bloodstream, not freely, they're bound. And so that's a tertiary. The one we looked at before, the classic one, is hemoglobin. It has 4 globin chains. Those are polypeptides. When you put all 4 of those polypeptides together and you stick the oxygen carrying group in the middle of them, you'll learn about that in blood, you have a quaternary structure. So if we have this jumble of stuff that's here, all making a protein, what happens when we alter temperature? What happens when we alter pH? What happens when we expose them to heavy metals? Think of something. The process is called denaturing. And what we do is we lose first the quaternary structure, we lose the tertiary structure, we lose the folding, and I'll do it the easy way with this. Secondary structure, the hydrogen bonds, and what we're left with, just the primary structure. The primary structure remains. Think A, okay? The amino acid sequence didn't change. What did we do? Went from a globular-based protein to the white. They go from that sort of liquid, and you heat them, you're denaturing it, and everything else. Again, when you're cooking steak or chicken, you're changing it from the inside. Look what happens. You begin to steadily denature it. That's all you're doing. You're making it into a form that's we are able to tolerate a little bit better and ingest a little bit better. And by denaturing it, and by definition, particularly with temperature, that is irreversible. We can do it reversibly. Think of the difference between freezing. Okay, some think we can defrost it. Sometimes if we freeze it for too long or improperly preparing it for freezing, we can create damage inside of it. That's less due to denaturing than the way you prepare it to freeze it. Okay. Commercially, when you freeze things in food science, there's a way of doing it to avoid problems with it. Pros will create, let's say, little cakes they're going to freeze. It's IQF, individually quick frozen. You put them on a sheet pan, stick them in a big freezer. Once they freeze, then they can package them without hurting. Unlike you and I, who just I got a bunch of extra stuff, throw it in a zip top bag and toss it in the freezer. Because the freezer goes. But what happens when if we froze it properly and we put a piece of chicken or a piece of meat in the freezer and took it out, within a reasonable period of time, it's not going to last forever. And you can always look up what the frozen life of those things. We take it out, defrost it, bang, ready to go. Probably won't taste a whole lot different, if at all. Most people would not be able to tell if it was done properly. So, as opposed to, you can't uncook the egg, you can't make the steak more rare. Pick an example that's silly, and it's all fairly obvious. That's denaturing. Thermal changes, thermal decomposition is, by its very definition, irreversible. What happens, it's just like us. Biologically, I do this again in micro. Here I think the living systems are something to what we call Velcro. You have midline, And that's what pH does. pH, the normal, let's say, is 7.4. Really, and if it's starting to change, going down that sort of slippery slope, as long as we're around there, we're good. Temperature's not a bell curve in living systems. Temperature looks like this. Starts as a bell curve, and then it's like a long cliff. 37 degrees Celsius is your optimal temperature. If you go to 40, if you go to 42, you're going to die. Add another 9 degrees to that, 98.6, you get to about 108. What happens? Proteins are small. That's why it's so darn important to understand this. So that's a little mini demo that's associated with this. So as we go back, 2 or more different polypeptides. Sometimes, typically, it's polypeptide groups. There's a lot of famous ones. You will see them. So things, test-wise, I will ask you, what is the primary structure? Okay, what do we find in a typical amino acid? That's what that is, an amino acid. Okay, that kind of thing. Secondary, what causes it? Hydrogen bonds. Tertiary, different types of factors, basically positive to negative, water soluble, fat soluble, all the stuff I wrote up there. Quaternary, 2 or more polypeptides. Proteins have 100 or more. Those are test questions. Okay, not going to have you draw the structure. Oh yes, this circle that's the peptide bond. Don't want to do that. Chemistry class, it's biochem. She's teaching it. They throw stuff at her. She's hard, I'm easy. We co-teach the MCAT students preparing for medical school in biology. I don't know. I don't know if this does anything. I'll see.
Shape. Each protein molecule consists of 1 or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
Oh, that was it? Well, that was dull. Fibrous. So we talked so before about globular. What you saw, that whole demonstration, me playing around with the paper towels, I'm going to play again. Globular. Fibrous. Strand-like. With one exception, water insoluble. The exception is called fibrinogen. You don't have to know that yet. The whole idea of the clotting cascade is to change. That's dissolved in your bloodstream. It changes it. Clotting cascade makes that fibrinogen change shape, become insoluble, forms like a fishnet, and traps red blood cells. Make blood clot. Very useful. You'll get that in blood. So these are structural proteins, a la the collagen, a la the elastin, the reticulin. You saw actin. Actin's an odd one. Myosin. Okay. Most have a tertiary and maybe a quaternary structure. Mechanical support and tensile strength, they are structural materials. Generally, with limited exception, they are water insoluble. When you heat them, they just get tougher. So and you've all, with something you're eating and you've cooked, it got to be well done. It gets tough, irregular, tough to chew. Maybe it's a part of what we call gristle. Yeah, not good eats. Okay. Single most abundant, that's collagen. You probably learned about that. I talked about the contractiles. They do this. So they're made out of those spirals. So individually, even I, in my weakened state, can do that. I'll even do that again. It feels so good. Oh no, don't look bad. I made it. But yes, that's right. If I take these and I braid them together, they're much stronger. Biologically, we braid. In surgery, we braid. When I would wire bones together, I wouldn't use a single piece. We use monofilament stainless steel when you do wiring of bones. What we would do is we take this long, it's about 3 feet long. You basically bring the ends together. My assistant would hold one end, I'd hold the other end, typically where the needle is, and we'd do the twist with a couple of hemostats, creating a braid. And then I basically would use that braid as a suture, drill holes in the bone and wire it together. I did do this stuff. Globular, much more functional. Enzymes, globular. Okay, all the time. Sensitive to environmental changes. Chemicals coming to them. What we used to think, you're going to see with enzymes in a second, we used to do something called the lock and key model, which is somewhat archaic. Now, lock and key would suggest that basically once the stuff that's coming in there, it bonds so closely. It really doesn't. It's sort of a close fit. Things make changes. As certain things come close together, we begin to see certain types of alterations occurring. So they're very sensitive to environmental changes, chemicals coming their way, acids, bases, temperature. They're either tertiary or quaternary. Okay, they have areas which we call active sites. So again, with the bad artwork coming. Saw it before. So effectively, it's big and it has these collaterals. That maybe when something comes along that's designed to shape it, we'll call it A. Another name we'll call these substrates. Another term you're going to see is called ligand. And when they come close to each other, they both begin to do a subtle change molecularly. Because of that, it facilitates them either coming, if there's more than one, they'll come together. If there's one in a full apart. That's the beauty. And then again, they're designed to be in a water-soluble system. Okay, antibodies, it's about their shape. You saw that Y shape surrounding that virus. Hormones, same thing, it's about the shape. Insulin is a hormone. That shape fits into an active site like that with a chain reaction. Hold this site here where your bond will be active. Don't want to go crazy with this. I don't know if it's in here, and if it's not, I'm not going to go nuts with it. It has to do with negative feedback. And a lot of times negative feedback relies on a different area of the enzyme. Unless they're pointing it out here in this presentation, I don't want to necessarily add it. So antibodies, again, antibodies basically circulate, and it's all about shape. Why that antibody? Was able to latch onto it. It's the shape. Our immune system tells it to make this little shape. That's called its active site. And that bonds to sort of a mirror image, okay, that's protruding from, it's called a protein spike, that you'll learn about in micro, that protrudes from that virus. Okay? And what it does, the antibodies, immense amounts of them circulate all over. They're just flowing, and when there's enough of them, sooner or later, they're going to contact the virus. They don't have eyes. They can't seek it out. There's no chemical attraction. It's a numbers game. Okay? And the more we have of them, okay, the more likely they're going to get that virus or that bacteria, and they're going to latch onto it, and boom, that's going to trigger the rest of your immune system to come and eat and gobble it up. That's how it works. It's not magic. It's all about chemistry, likelihood of, that's where temperature comes in, increases the likelihood of collisions. That whole active, average kinetic energy, more likely to slam into each other than they attach. That's how your immune system works. It's extraordinarily complex. I'm trying to make it, and maybe this helps, maybe it doesn't. I'm trying to, sort of, you're stuck with me, so you got to get my spin on it, so to speak. This is how I've tried to understand it over the 50 years or so I've been working with it. Both teaching it and then practicing it. So you get lucky and have somebody else who's more interesting. And they probably will not show you pictures of the dog, the cats. Oh, the cat's such a pain in the ass. Do you know what that cat did? We left the door open because he likes to go out. We were away for 4 days. Nowhere to be found. We got back yesterday about 1:30. Cat, my wife's having it. Just really upset that her cat, because he's been across the street. And maybe you have a cat like that. I looked all over the house. Maybe he's hiding because he gets indignant because we weren't there to take care of him when he wanted us to. The cat rule. The other cat was fine. Beat him, he's happy. My wife released me a note this morning because she's already asleep. She goes, I went upstairs to go to bed at midnight. He's sitting on the bed. I have no idea how he got there. The door was open the whole time. Never saw him come out. This morning at 5:30, he's mewing at me. I want treats now. That's what I have, and I think my kids are better. Moving on. So you saw a wonderful animation. Antibodies, hormones, chaperones, just what the name says. Basically, they escort molecules to come together. So that's called a molecular chaperone. Nothing new under the sun with that. And here's the denaturing nugget. Fibrous proteins are more stable. Active sites become deactivated because all that happens is you change the shape. When that temperature starts to occur, what happens is it causes that spot to begin to narrow. And now that can't fit in there. That's what shape is. Long shape. Causes those antibodies to begin to break down. They can't latch on. Think of any one of those examples that are there. The chaperones can't latch onto the molecule and move it. Typically, increased pH, more common, and increased temperature. They are much more common than the opposite. Okay? Usually reversible if we are able to reverse it quickly. Irreversible. Okay, and again, we have cold is much more forgiving, okay, than heat that are there. And here, globular proteins that act as catalysts. They're everywhere. They can catalyze. It's a fancy term we use for how they work. Thousands of reactions in a split second. All they do is they increase the speed of the chemical reaction. The way they do it, every biological reaction requires energy input. You have to put energy into the system for it to work. So there's always like a startup type of thing. You got to turn in the old days before you had push button cars. And then you have to push the button of the car to get it to start. Different. The big question is you learn about with energetics, whether it releases energy and breaks bonds overall or stores energy has to do with how much energy you got out. You're always going to put some in and you're always going to get some out. The question is, how do they balance? Okay. These would take forever. Activation energy. I mean, this paper towel. If you came back tomorrow, is it going to be a paper towel like this, assuming nobody moved it? Next week? Next year? Sure. 100 years from now, what's going to happen? Is it going to look like this? No. If you go to the library and find one of those old books you opened and the bug comes out, those books, the stacks we used to have in old libraries. I mean, it looks like a bad movie. Okay. The moisture in the air itself will begin to denature the proteins, denature the structure that are in here over an immense amount of time. Biologically, we don't have that time. That's what catalysts are. And by definition, this is important, lowers the activation energy. So as you have millions of reactions per minute. So, and effectively that increases the speed. So temperature may increase the number of collisions, but now we're doing something to lower the need for the enzyme that are there. Let me see if I can get this. Yeah. We'll do it this way. It's sort of like this. For instance, we look at the energy balance. We put in energy, but in this case, we got a certain amount of energy, and we add up the amount we put in and the amount we obtain, and it's either going to be we're either going to put in some and get more out. That means we broke bonds, or we're going to put in some and get less out. That means we created bonds. In any event, we have an energy of activation. This just lessens it. That's all. I mean, it's the old. It's if you go skiing, I don't. I mean, imagine how hard it would be to walk your way up, to get to the top of the mountain, so to speak. A ski lift basically lowers the activation energy. That's the best way to describe it. I mean, whatever works for you works for me. So lowering the activation energy is what happens. This is the other thing. Most of them, we have different things that work with it. I drew a generic form of an enzyme. Most of them require other stuff that we consider essential. They're called cofactors and coenzymes. So the APO enzyme, fancy name, is all the protein. Cofactors, by definition, are inorganic. They're typically metals. We have a great name for them, minerals. Coenzymes are organic. We call them vitamins. They are needed for the most, not all of them require both. Most of them do. So, for instance, for insulin to function, it probably needs a tiny amount of zinc. So there's all, hemoglobin requires a tiny amount of iron. Thyroid requires a tiny amount of iodine, for all of those things to work. So the difference, what the definition of cofactors and coenzymes, inorganic, mineral, organic, for our purposes, vitamin. They're very specific. One enzyme that breaks apart particular protein for something will not necessarily break a different protein. It has different R groups. They're very, very specific. Enzymes, I mean, you saw it before when we were looking at the sugars. In order to create or break down table sugar, we have sucrase. We talked about lactase. The absence of that, which we were born in the absence of that, makes us lactose intolerant. We already discussed that. So the ASE is the substrate, hydrolases or oxidases, different things. One involves water, one involves oxygen. Actually, it has something more to do with oxidation reduction reactions, and we don't like them. So they lower the activation energy. Okay. They want to say priming the pump. I'll buy that. Proceed quickly at body temperatures. Substrate, I wrote up there, bind, it complexes, it changes in some way, shape, or form, and it's rapidly released. Voila. Bind. And here, 2 amino acids bind, come together, peptide bond. So that would be a peptidase. ACE doesn't mean it's going to break it apart. It means it's involved. There's lots of different peptidases. And we'll stop it here because we'll do DNA and RNA and finish this chapter tomorrow and go back and start looking at cells, but I'll beg your pardon on Wednesday. So thank you. Have fun. Glad to be back.